Assalamualaikum and a very good morning to all panel and my supervisor. Today I will present about my FYP proposal. My name is Wan Muhammad Azim Fikri bin Wan Muhammad Abdul Malik. My metric number is S55115. My program is Bachelor of Aquaculture and my supervisor Dr. Sharifah Nur Emilia binti Syed Jalal Mifada. The title for my FYP project is Effect of Depuration Process on Microbial Population of Oyster Mangalana Iredale. As the introduction, in the background study, I will present generally about my FYP project. My species name is Mangalana iredale. It's the one of the oyster species that live in seawater. It belongs in family Ostridae. The Mangalana iredale name that renamed for Casotria iredale. Mangalana is the parent's name for Casotria, so acceptable to use Mangalana iredale as name for oyster species. However, the focus of the project is the contamination of the microbiome that consists in oyster, such as virus and bacteria. So to solve that, apply the depuration process that function to filter the contaminant from oyster. The system that will be used is flow-through system. Moreover, this is to enhance a good quality of seafood safety for the consumer. In next slide, for problem statement. The problem is microbiological pollutant in water. As the filter feeder, oyster will filter all food in the water. They will eat many microorganisms that live in water. However, they may expose to toxin and microbial contaminants. When humans eat the oyster by consist with the bacteria and a virus, it may affect to consumer and get the disease. Example of disease for virus is gastroenteritis and viral hepatitis, and for bacteria disease is typhoid. In part of the significant study, there are 4 things, which is purification by using seawater to isolate the contaminants from oyster, UV light as the infection method in depuration, eliminate the possibility of sickness or death to ensure that the community has access to healthy seafood. Then, for the objective, the main objective of the experiment are to observe depuration impact of microbial population on oyster Mangalana iredale. In method and material, first, sample collection. The oyster sample will be collected at Seti Wetland Terengganu. Second, oyster depuration. Flow-through system to conditioner tank, record water parameter, analyze in 3 times in 10 days, which is first day, third day, and test day. 3. Experimental design. Triplicate 5 liter commercial bus, 150 gram per sample with 30 sample of oyster for each flash. 150 ml of water used. Oyster muscle part and outlet water used to analyze microbial population. Number 4. Aerobic mesophilic counts or APC, used to count an aerob bacteria. Aerobic mesophilic counts incubate at 30 Celsius for 2 days. Psychotrophic counts incubate at 4.5 Celsius. Determined on Triptychus soy agar plus 0.5% and ACL both the method. First, for the Pseudomonas, incubate at 25 Celsius for 2 days. Determine Pseudomonas number on Pseudomonas agar base. Fusidin, cetramide, and cephaloridin as supplement in agar base. Second, Verbio SPP, incubate at 30 Celsius for 2 days. The assumption count of Verbio SPP on thiosulfate citrate bile sucrose agar. And lastly, for Aeromonas SPP, incubate at 28 Celsius for 2 days. Determine the presumptive of Aeromonas SPP on starch ampicillin agar. Number 5. Lactic acid bacteria or LAB. Process incubate at 25 Celsius for 3 days. Count on available plates of Demon Rogosa sharp agar. And lastly, for number 6, most probable number NPN. This is a method used to estimate the viable number of bacteria in a sample by inoculate broth. So this method by doing incubate at 37 Celsius then 44 Celsius. Beta glucuronidase positive, Escherichia coli counting, liquid medium culture technique and NPN calculation. In statistical analysis, the number of microorganisms per gram convert to log CFU per grams and express. The results are shown as mean standard deviation or SD. Statistical association and the importance of variation in bacteria counts means and use the ANOVA method for the calculation. In expected result, in the experiment that will be held, I expected that population of microbial in the oyster Mangalana iridale will undergo reduction. The experiment will running in 10 days. In first day, due the depression process of the oyster, the population microbial undergo reduction at master part. After that, in day 3, the microbial in oyster increase of reduction. At the 10th day, more reduction occur. So the population of the microbial in an oyster will increasing day by day. So this is my references. My Gantt chart. So that's all from me. Thank you.